Right, so if you remember from the previous lesson, we spoke about the requirements for photosynthesis and the products of photosynthesis. And then we spoke about how and when during the light phase and the dark phase these requirements are used up and the products are made. If you recall, the two products of photosynthesis was glucose, C6H12O6, and oxygen. Now, oxygen was made as a product, and if you recall correctly, it was released in the light phase into the atmosphere. It's a byproduct of photosynthesis. In other words, it's a waste product. It's not the essential product. I said to you in the previous lesson that trees do not undergo photosynthesis because you need to breathe oxygen. Oxygen just happens to be its waste. The point of photosynthesis is to make glucose, C6H12O6. That glucose that gets made by the plant gets stored as starch, which we learned in the previous lesson too. So, if I want to know if a plant has undergone photosynthesis, I am always going to look for starch. This should take you back all the way to grade 8 when you did the starch tests and you did iodine on potato and cheese and bread and those funny things. We are always going to be looking for starch to prove that photosynthesis has taken place. So in saying that, we are going to do a number of experiments that will prove photosynthesis has taken place. But it's not as easy as, is there starch? Boom, yes, there's photosynthesis. No, we're going to test factors. So for example, if there is carbon dioxide available, will photosynthesis take place? Which we know the answer will be yes. But will photosynthesis take place if there is no carbon dioxide? Or will photosynthesis take place if there was no radiant energy? We're going to test those, and ultimately then we'll test for starch. It's a little bit more complicated than just testing for starch each time. So let's get to it. And we're back to being a novice. Fun. Okay. Right, so before I get into all the hectic detail about the different factors that we can test, I'm going to just talk about how we test for starch. So this is revision of grade 8 work. Sorry about that little break in there. I don't know why it happens to me every single time, no matter how many times I've done these videos. Right, so if I'm going to test for starch, I know that I'm testing for the stored form of glucose. I'm first going to take a leaf. Remember, a leaf is where the chlorophyll is contained. I'm going to take that leaf, I'm going to boil it in water. When you boil the leaf, you are breaking down the cell wall. Remember that a plant cell has cell walls, and cell walls are not permeable. They are we can't have oxygen and carbon dioxide leaving the cell wall. Remember, they leave and enter via stomata. So we need to break the cell wall so that we know that eventually we'll have the penetration of the alcohol or the iodine, etc., that we're going to use. All right, so that's step 1. Secondly, we are going to put that leaf that we've already boiled in a test tube that contains ethanol or alcohol. That is going to dissolve the chlorophyll, break down the chlorophyll. So in other words, it's going to take the green out of the leaf. So if you think about it, if I have a test tube with ethanol inside it, that ethanol is clear. Once I've put my leaf that has been boiled inside that ethanol, once I've left it inside there for a little while, I'm going to find that the ethanol becomes green. It pulls the green, the chlorophyll, out of the leaf. Then I'm going to remove that leaf and I'm going to wash it in cold water. Okay, when I wash it in cold water, I'm getting rid of the any alcohol or ethanol that was left on the leaf, and I'm going to soften it up again. It goes quite hard when we dissolve it or when we put it into the ethanol. That leaf, that soft leaf that I've rinsed now, I'm going to place in a petri dish. I'm going to take some iodine, which is the indicator for starch, and I'm going to pour some droplets of iodine onto the leaf. What we find is that if starch is present, so in other words, if photosynthesis took place, the leaf will turn a blue-black color. If there was no starch present in the leaf it doesn't actually change color. The iodine is originally a reddish-brown color. The iodine that drops onto the leaf will turn the leaf a reddish-brown, and it won't change. Okay, so in other words, there's no change in color. When there's a positive result, the reddish-brown color of iodine becomes blue-black. The leaf will be stained to that color because we removed the chlorophyll. Okay, so if I was going to do an experiment and ask you questions here, the aim would have been to determine whether starch was present in the leaf. Okay, and your result shows that starch is present in the leaf or starch is not present in the leaf. But if I said to show whether photosynthesis, aim to show whether photosynthesis took place, right? Because the result, or if the result said the leaf turns blue-black, then the conclusion would be photosynthesis did take place. If the result was that the leaf remained or turned reddish-brown, then the conclusion is photosynthesis did not take place. Right, you need to remember this for the rest of the experiments. Now, there is no use, grade 11s, of testing a leaf. Okay, I can't go into my garden right now, pick a leaf, and say, I'm going to decide to test this leaf to see if photosynthesis took place. Great. What am I going to find? I'm going to find that definitely 100% photosynthesis will have taken place. If my leaf is green and my plant is growing, photosynthesis has taken place. I cannot use that leaf to prove anything. It could have been photosynthesis could have taken place because of carbon dioxide, or some person could have just lied to us and maybe we just believe it. So in order to prove anything, I must first take the starch out of a leaf and then expose that leaf to the conditions that I want to see if photosynthesis took place. Okay, so in other words, we're going to take a leaf from our garden, and we need to now first remove the starch from that plant. Now, similar to how humans work, if you haven't eaten for a number of days, you know that you're going to, when you eat, you store any excess energy that you have in your liver, and that excess glucose gets stored as glycogen. And when you do any physical activity, you burn off that glycogen. If you are Bear Grylls and you are in some kind of desert jungle thing for a number of weeks, then, and you are hardly eating, then your body burns off that glycogen. So think of anyone playing Survivor. They store lots and lots, they eat lots before they go, and they store a buildup of glycogen in their liver. And as their days continue in Survivor, they're eating less and less. So they're losing so much weight because they're burning that glycogen store that they had off, and eventually they don't have a glycogen store left, and so they start losing weight. All right, so in the same sense, remember that glucose stores as starch, and that is the plant's version of stored energy. So we need to make this plant lose its stored starch. So how we do that is we take a plant, and not a leaf, a whole plant, because we don't want the leaf to die. I'm going to take a plant and put it in a dark space, a dark cupboard, for 48 hours. What does it mean if I say it's dark? Then it's not being exposed to radiant energy. So in other words, from the minute I've put it in the cupboard, it cannot continue light phase. So it cannot continue to do photosynthesis, but it will have energy stored from its previous rounds of photosynthesis. So we're going to leave it in the cupboard, knowing that no further photosynthesis can take place, and then leave it there for 48 hours so that the glucose that was stored as starch is broken down. So that starch will then be broken back into glucose. The plant will keep using that glucose to keep doing its metabolic activities until such time that the plant runs out of that energy, and then it will die. So that's why we don't leave it for 6 weeks in the cupboard, because then we're just going to have a dead plant, which is not what we were looking for. So we leave it for 48 hours, which is just enough to remove the starch from the plant. How did it get removed? We stopped photosynthesis from continuing, and we broke down the starch that was stored into glucose, which then continued to use the plant continued to use that glucose for its metabolic processes. So just to put it into human terms for you, it's you do not eat, okay, you do not eat, and while you do not eat, your stored glycogen in your liver is being broken down, right, up to the point where there's none left, okay? That's when you will start losing weight. That's when the plant will start to die, all right? Okay, so we know that if we've taken the starch out, then now we can use that plant to determine different factors. Okay, so we're going to use that information repeatedly. So please just keep all of that in the back of your mind. So it's not the first experiment, but it's the first factors experiment that we're going to do is to talk about proving whether radiant energy, light, is necessary for photosynthesis. To prove that light is necessary, we are first going to have to de-starch the plant. So now, in a test, you would say de-starch the plant. You wouldn't have to give me the steps unless I specifically asked you how to de-starch a plant. Okay, I hope that makes sense. So, when we talk about is light necessary for photosynthesis, we're going to say aim to test whether radiant energy or sunlight is required for photosynthesis to take place. Okay. Hypothesis. What do you think is going to happen? Radiant energy is required for photosynthesis to take place. Method 1. De-starch a plant. Okay. We have to de-starch the plant. I told you just now. We have to de-starch it to be able to start off with a control. If I'm starting off with a leaf that already has photosynthesis, how am I proving anything? Okay, so I'm using my de-starched leaf as a control. Then I'm going to take one of the leaves from this plant and cover part of that leaf with tinfoil. So here you can see what they've done here. I'm going to cover the part of the leaf with tinfoil and leave another part of the leaf exposed to sunlight. I'm then, step 3, going to allow the plant to photosynthesize. How am I going to do that? By putting the plant in sunlight. Okay, I'm testing sunlight. So I can't now put it into a dark cupboard. I've got to put it into sunlight to be able to expose it to the factor I'm trying to test. Okay, now what's going to happen is the following. There's my leaf. This is the part of the leaf that was covered in tinfoil. Okay, and this is the part, these two parts here where the dots are, were exposed to sunlight. Now, I took all the starch out of the leaf originally. Okay, so wherever light wasn't able to penetrate the leaf, there should be no starch. But in these areas, this area with the dots here and this area with the dots here, those were exposed to sunlight again. And when they were exposed to sunlight again, they should have photosynthesized, or that part of the leaf should have photosynthesized. And so therefore, we'll see that if I take iodine and I perform the starch test, so that same leaf that I've now covered with tinfoil, I'm going to take that leaf, pick it off my plant, boil it, alcohol, blah, 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 iodine. I'm going to put iodine on the area where there was tinfoil, and I'm going to put iodine on the area where there was no tinfoil. On the area where there was no tinfoil, it's going to remain reddish brown. On the area where there was no tinfoil, I hope I said that right. On the area where there was tinfoil, it's going to remain reddish brown. On the area where there was no tinfoil, the area that was exposed to light, it will turn blue-black. That will show that where the leaf was not exposed to light, no photosynthesis occurred. And where the leaf was exposed to light, photosynthesis occurred. And so if we look at my original aim, which was to test if light is necessary for photosynthesis, I can answer it by saying light is necessary for photosynthesis to take place. Okay, and therefore I would accept my hypothesis that I made. Okay, the next experiment is carbon dioxide necessary for photosynthesis? So now again, remember that to do the control, we must use a plant that has been destarched. Okay, I've explained that, so I'm not going to re-explain it. Now I'm testing for carbon dioxide. I'm wanting to know, is carbon dioxide necessary? We know it is necessary. So if I had to do this in an experiment, in a write-up, aim to test whether carbon dioxide is necessary for photosynthesis. Hypothesis, carbon dioxide is necessary for photosynthesis to take place. Okay, now in this, just like in the previous experiment, I'm going to go back here, I had a control, right, and a test. I was controlling it at one point and I was testing it at one point. I'm going to again need a control and a test. That means I'm going to need an experiment where I'm using a destarched plant and not exposing it to carbon dioxide, and I'm using a destarched plant and I am exposing it to carbon dioxide. Same as what I just did in the previous experiment. So in this experiment, I'm going to set up a bell jar. Inside the bell jar will be my plant. This plant, if you think of a bell jar, okay, if you think of just a M&M inside a cup. I put an M&M down on my table and I take a cup and I turn it upside down and I put it onto the M&M. Is carbon dioxide surrounding that M&M? Yes. Okay. And now we don't want that carbon dioxide. We need carbon dioxide to be removed to be the control. So these two things are very important here. When I set up my control, my bell jar, I have to, one, use soda lime. Please, this is important. Soda lime. I have to use soda lime in the entry point, in like a straw tub in the top of the jar to prevent the entry of carbon dioxide. So I don't want any excess or extra carbon dioxide to enter. So the bell jar, when we set up this apparatus, obviously in the test over here, I'm going to have an entry point for carbon dioxide. So I need to seal this. I can't just close it off. I need to just prevent the entry. So I'm going to use soda lime, which is going to prevent the entry of carbon dioxide. But now, just like I said about my M&Ms or my cup with my M&M, I already will have carbon dioxide inside this jar floating around here. So I need to get rid of that carbon dioxide that's floating around here. And how I do it is I put a cup next to my plant that contains sodium hydroxide. Sodium hydroxide absorbs carbon dioxide. In other words, this carbon dioxide that's floating around here in the air is going to be absorbed by the sodium hydroxide to remove it from the air, so that eventually there is no carbon dioxide inside this bell jar in order to do my test. Okay, this setup over here is my control. I am going to expose it to sunlight because I'm only testing carbon dioxide. I can't test more than one factor at a time. All right, my test then, obviously carbon dioxide needs to be able to enter my bell jar, and I'm going to put a jar there because I did in the previous one. But in this case, I'm just going to put water in it because I don't want the carbon dioxide to be absorbed, right? This here is being used to absorb carbon dioxide. Now I actually need the carbon dioxide to do my test, so I'm just going to use water as like a placebo, basically. Right, so my control is my plant being exposed to no carbon dioxide. My test is my plant being exposed to carbon dioxide. Now, please, guys, in the question, you need to know these two things. Soda lime prevents the entry of carbon dioxide, or sodium hydroxide absorbs the carbon dioxide from the bell jar. Okay, those two things are very important to know. Control, again, and test, again. All right. Okay, so I see I don't have a follow-up on the slide, but it's easy enough. So once I've done this, what am I going to do? I'm going to take a leaf from this plant. I'm going to take a leaf from this plant. I'm going to do the starch test. When I do the starch test, the leaf from this plant will stay reddish-brown. The leaf from this plant will turn blue-black. So it will prove that photosynthesis happened in this plant and photosynthesis did not happen in this plant. And therefore, I can conclude that carbon dioxide is necessary for photosynthesis. Right. Okay, next one. Is chlorophyll necessary for photosynthesis? So again, I'm going to destarch a plant. The important part here is this, that it says a plant with variegated leaves. What does variegated leaves mean? This is a variegated leaf. And you see that the outside of the leaf is yellow while the inside is green. Where there is no green, there is no chlorophyll. So this yellow part of the leaf, it actually doesn't contain chlorophyll. And that's important because by using a variegated leaf, we are able to test where chlorophyll is present and chlorophyll is not present. Okay, so we're going to take that variegated leaf, we're going to destarch it, firstly. Secondly, we are going to then take that, again, we're not going to take the leaf, guys. So I keep saying leaf, but understand that I mean a plant, because if I just take a leaf, it's just going to die. So I'm going to take a plant, a variegated plant, destarch it. Then I'm going to take that plant that I've destarched after 48 hours and now put it in the sunlight so that it is allowed to photosynthesize. Okay? When I'm done that, when I've exposed it to sufficient sunlight, I will pick a leaf. I will then perform the starch test, as you guys know how to do already. When I test for starch, I'm going to find that where there was green, so this one is the opposite, okay, I just want to show you. But where there was green, the leaf is going to turn blue-black. And where there was no green, the leaf is going to stay reddish-brown. Okay, so just that picture and this diagram is a bit opposite. Here it shows that the inside is non-green and the outside is green. So therefore, if I put iodine on the non-green part after I've done the starch test, or after I've done the boiling and alcohol, etc., then this part over here is going to remain brown. The outer part where there was green, where there was chlorophyll, is going to turn blue-black. And therefore that is going to prove to me that where the green part was found, photosynthesis occurred. And so therefore we can conclude that chlorophyll is necessary for photosynthesis. At this point, just because these diagrams are what we get, but black and brown are not good enough. Blue, black, and reddish brown are your words to use here. Please make sure that you write that down or that you remember that. Brown and black are not good enough as the colors. When iodine tests positive for starch, it turns blue-black. When it indicates negative for starch, it remains reddish-brown. Okay, so here's my example then of my variegated leaf. Here I had chlorophyll. On the outside I had no chlorophyll. When I do my test, where the chlorophyll was present, it will turn blue-black. and where there was no chlorophyll present, it will remain reddish brown. Okay, now, so those are experiments to test if chlorophyll is necessary for photosynthesis, if carbon dioxide is necessary for photosynthesis, and if light is necessary for photosynthesis. Okay. You learned in grade 8 about lime water. Now, please do not confuse lime water with soda lime. Okay. Soda lime, we know, was now used in the second practical, the second experiment, to remove or absorb carbon dioxide from the atmosphere, from the air. Okay, and it's a mixture, as you see here, of sodium hydroxide and calcium hydroxide. Lime water is an indicator. If I blow with a straw into lime water, when I remember that as a human, I am undergoing cellular respiration, so when I'm blowing with a straw, I blow out carbon dioxide. So when I blow out that carbon dioxide into the lime water is going to turn milky. Okay, lime water is originally clear. It's going to become milky. That is going to indicate that carbon dioxide was present. But lime water is not the same as soda lime. All right, soda lime removes carbon dioxide from the atmosphere or from the air. Right. And next experiment is oxygen needed. Not needed. Is oxygen produced during photosynthesis? So every time we do the starch test, we're obviously proving that starch, glucose, is made when photosynthesis occurs. Now we need to prove that oxygen is in fact a byproduct of photosynthesis. Okay, so what we're going to do is, if we have a setup where we have a leaf or a plant again that is going to undergo photosynthesis, we cover that plant with a funnel. If that plant is making photosynthesis, listen to me, I can't speak today, is undergoing photosynthesis, we know in theory that it's releasing oxygen gas. So we are going to cover the plant with a funnel and then cover the opening of the funnel with a test tube. What will collect inside that test tube is oxygen, right? Because we know that oxygen is the byproduct. When I take that test tube and I expose it to a glowing splint, the glowing splint is going to ignite. A glowing splint is an indicator for oxygen. So I want you to think of like a little matchstick that ignites if it's exposed to oxygen. Okay, so it's not like a matchbox where you've got to take the match and strike it against the matchbox. It is literally, if I expose it to oxygen, it's going to ignite. So in this case, when I take this test tube and I expose it to a glowing splint, the splint is going to light up, showing that oxygen did in fact take place. Okay, and that is how we prove that oxygen was produced during photosynthesis. Okay, so if we've now spoken about all these experiments, we know that those are all requirements for photosynthesis. And therefore, if they're requirements for photosynthesis, then if they are not available, then photosynthesis cannot take place. We know that those factors those requirements can in fact be affected by a number of factors. And those factors can affect the rate of photosynthesis. So just before I even get anywhere, think of a plant that is exposed to direct sunlight as opposed to a plant that is in a cooler, shadier area. Photosynthesis is going to be different because of different factors. Right, so the first one is light intensity. So sunlight radiant energy is a requirement for photosynthesis. If a plant is exposed to maximum light intensity, the rate of photosynthesis will increase. But there is an optimum. Okay, at a point it can be too much. But as the intensity of the light increases, so will the rate of photosynthesis. What they like to ask you in tests is like an example of a plant being moved closer to a lamp. So if I put a lamp and I have 60 centimeters, I have a plant growing away from that lamp. And then I move that plant, or I have another plant growing at 30 centimeters away from the lamp, and then I have another plant growing at 10 centimeters away from the lamp. I'm going to find that the plant growing the closest to the light is going to have the best rate of photosynthesis because it's getting the best light intensity. Okay, so you need to know for test purposes, light intensity increases the rate of photosynthesis. As light intensity increases, the rate of photosynthesis increases. At this point, I need to remind you, if I recall correctly, with my classes, I did just before lockdown, we went over that practical that you guys did in term 1. And I said that we do not, in life sciences, say it is directly proportional or indirectly proportional. That is a maths answer, and I want you already now in grade 11 to scrap that from your bio knowledge. You do not have, or you do not have to, you must not say it is directly proportional or indirectly proportional. You will not get the marks in matric. You must say, as this one increases, or as this one increases, this one decreases, or whatever the case may be. All right. The second factor is the concentration of carbon dioxide. So this one's kind of obvious. If carbon dioxide is necessary for photosynthesis to take place, then more carbon dioxide would mean more photosynthesis. Okay, so in an area where the plant is exposed to more carbon dioxide, the rate of photosynthesis is going to be better. The next one, temperature. The higher the temperature, the better the rate of photosynthesis will be, but only up to a certain point. I want you to think of grade 10 again and the denaturing of enzymes. Okay, photosynthesis is controlled by enzymes. So if I denature those enzymes, suddenly it's denatured, it cannot continue. All right, so our rate of photosynthesis will increase up to the optimum temperature. Right. And then lastly, the chlorophyll concentration. It says a lack of leaf chloroplasts will decrease the rate. So in other words, more chloroplasts, better rate of photosynthesis. Okay, so in other words, actually all of these, light intensity, carbon dioxide concentration, temperature, and chlorophyll, you can say that as that factor increases, so does the rate of photosynthesis. Except you must remember, especially it always up to an optimum, and in this case there will be denaturing of the enzymes, then the photosynthesis will stop. Okay. Right. Grade 11s, that is the end of photosynthesis then. I'm going to just take this opportunity while I have your attention and you're listening to a video to say, I freaked you all out by saying that there's going to be a test, and I had a lot of you private message me and say, ma'am, what's going to be in the test, blah 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 blah, because unfortunately the way you guys are wired, the minute we say test, you go into a flat spin. The point, as I try to explain to you, is not for me to say test and you quickly in a week or 5 days cram all of this information that I've taught you throughout lockdown into your head so that you can pass a test. That's not the point. Okay, we're not at school. That's what most of you do when we're at school. I did it when I was in school too. But there's a difference. The difference here is, once we're back at school, we do not know how things are going to be or when we're going to be there. What we do know is that we have further work to do. won't really have the time, or much time in any case, to revisit this work. So while we will revisit a little bit of it, I will expect that for the most part you've done the work, so that I can refer to, let's say, for example, I say sodium hydroxide, you should know what I'm speaking of without me having to re-explain it. Therefore, I'm going to do an assessment. I will send you the assessment. You will do it in your own time. Okay? There won't be a due date. The due date will be, say, by the time we come back. You'll have to have done it by the time we come back, but who knows when that will be. So it's not going to be, I send you the test today and I want you all to do it. I will say within give you a week to do each topic or whatever the case may be. It's obviously open book. You can use your books, but the point isn't to get 100% and not know the work. The point isn't to go find the answer on Google or to find the answer in your textbook. The point is for you to know, do I actually understand this work? And to be writing down what you do not understand. Some of you have been very proactive in this time, sending me messages and saying, ma'am, I don't understand this, or what is this, or how does this work, or please just clarify this. That's great. I'm here for that. Okay, keep doing that. If you have further questions or you need further clarity, write it down so that when we're back, you can come and ask me and we can fix those things up. But the point, grade 11s at this point, is to make sure you are covering this knowledge, this content, that it's going into your head somehow a little bit, so that you don't fall short when we are back. To give you an example, any student who has not had access to the internet over this time, that student is going to be given all this information, same as you, and told to catch it up, and it's going to be very hard for them. And while I will be there to support them, definitely 100%, my time is going to be required to help those people because they genuinely didn't have access to these things. My heart would break if those of you who do have access and are receiving these messages and are receiving these videos and are choosing not to work, those people, my heart will break for you because you are going to be expected, just like everybody else, to catch it up. Okay? So don't freak out about a test. It's not a test. It is to ascertain your knowledge, ascertain your understanding, so that we can make sure that you are where you should be before you write your final exams. Okay. And then also just a little bit of a message. And I'm speaking out of turn here, but I want you to not stress grade 11. Some of you are stressing so heavily about grade 11, and grade 11 being the year that they use for marks for varsity and this and that and the next thing, and how are you going to pass this year, and if you are going to pass, it's not going to be so great, your marks aren't going to be so great because it's self-study, da-da-da-da-da-da. I want you to know, in Mr. Lawrence's words, talking to us as staff, we are operating under abnormal, unusual circumstances. The government, the universities, etc., are not just going to use your grade 11 mark blank like that and say you weren't good enough. Okay, plans are going to be put into motion. We don't even have any idea of what's coming for us, when you'll be back, if you'll be back. We wait for that kind of information. So I want to put your minds at ease, grade 11s, to say, whatever happens, know that your future is not doomed because of coronavirus, because of lockdown. Your futures are just as bright as they were before this all happened. And as long as you continue to work through this, as long as you continue to do what's been expected of you, you are doing the most that you can. Remember, there are schools, 90% of South African schools, kids, have not done any work during lockdown because their schools do not have the infrastructure. Their teachers do not have the infrastructure. Their kids do not have the infrastructure to be learning. So at whatever point, and especially obviously for matrics, but everybody is going to have to be on the same page when we write exams. Know that this work that we've been doing has been for your best benefit to make it easier on you when we do go back. But also know that it's not going to impact your life, your plan, your future, your career that you have set for yourselves. Just keep your head up. Keep swimming. I promise you, I'm scared shitless too some days. And we'll all be okay. All my love, and good luck with your studying.